Hello dear students, our today's topic is from Unit 2, Principles of Animal and Population Genetics, practical number 19, and title of the practical is Calculation of Gene and Genotypic Frequencies. First of all, what is the meaning of population? Population is a group of interbreeding individuals. It may be a breed, species, or group of individuals present in a particular area at the particular time. So what is the meaning of genetic constitution? Genetic constitution of population may changes from one generation to the other. The basic constitution of the population is the gene, and as we knows that genotype does not transmit from one generation to the next generation, as genotype break down during gamete formation, and genes which are present and that in that particular gamete get transmitted from one generation to the next generation. So genotype does not transmit from one generation to the next generation, but genes get transmitted from parent generation to the progeny generation. First of all, what is the meaning of gene pool? Gene pool is total genes possessed by a male and female of that particular population. So here we are considering totally a breeding male and female, with the male and female which are mostly breedable. So according to the genetic constitution of our population, gene frequency and genotypic frequency are the most important criteria. What is the genotypic frequency present in our population, what is the genotypic frequency present in our population, according to that we are able to know whether there is a variation for that particular gene or not. So first of all, what is the genotypic frequency? So genotypic frequency is the proportion or the percentage of the genotypes in the population. Here we are considering a single gene which is present at a fixed locus with the two alleles. One is dominant and another is recessive. Here we are considering capital A is the dominant allele and small a is the recessive allele. When we consider a single gene with two dominants, we have possible three genotypes, that is homozygous dominant, heterozygous, and homozygous recessive. So when we are going to calculate a genotypic frequency in the population, first of all we know what is the genotypes present for that particular gene in the population. So here we will have three different genotypes in our population of the frog. There are 15 number of individuals present in our population. So first of all, what is the genotypic frequencies which are obtained from the number of different genotypes in the population or in a sample of that population. So number of homozygous dominant, that is capital A equals to DN. Number of heterozygote dominant, number of heterozygotes, that is capital A small a, it is indicated by HN, and number of homozygous recessive small a, and it will be indicated by RN. Here the total number of individuals present in the population we are considering, or we are indicating it as a capital N. So capital N equals to number of homozygous dominant genotype plus number of heterozygous genotype plus number of homozygous recessive genotypes. It is also given as DN plus HN plus RN. So frequency of homozygous dominant, that is capital A is indicated by capital D. Frequency of heterozygous, that is capital A and small a, is indicated by H, and frequency of homozygous recessive small a is indicated by R. So the frequencies of different genotypes in the population are obtained by dividing the number of individuals of a genotype by the total number of individuals in the population. It means that if we want to calculate or to know the frequency of homozygous dominant capital A, then we are going to count the total number of capital A individuals in our population, and it is divided by the total number of individuals present in the population for that particular gene. So first of all, frequency of homozygous dominant. It is indicated by capital D. Capital D equals to number of homozygous dominant, that is capital A upon total number of individuals, or it is also given as dn upon n. For heterozygote genotype, H is equal to number of heterozygote genotypes, that is capital A small a, divided by total number of individuals. And the frequency for homozygote recessive genotypes, that is small a, it is indicated by R equals to number of homozygous recessive genotypes present in the population upon total number of individuals. So the sum of the genotypic frequencies equals to frequency of capital AA plus frequency of capital A small a plus frequency of small a equals to 1. Or it is also given as D plus H plus R equals to dn upon n plus hn upon n plus rn upon n equals to 1. Remember that frequency of homozygous dominant D, frequency of heterozygous dominant H, and frequency of homozygous recessive R, it always remains 1. So next is calculation of gene frequency. Here our total number of individuals in the frog population are 15. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So we will have 15 frogs. So frequency for capital A, that is homozygous dominant, equals to frequency of AA upon N. So Dn equals to our 8 means here 1, 2, 3, 4, 5, 6, 7, 8. So total number of homozygous dominant genotypic frogs are 8 upon total number of individuals present in the population are 15. So it equals to 53. Next one is heterozygous. Frequency of heterozygous genotype, that is number of heterozygous individual upon total number of individuals. So we will have 4 heterozygous individual, 1, 2, 3 and 4. So 4 upon total number of individuals, our total number of frogs are 15. So 4 upon 15 equals to 27. Next one is frequency for homozygous recessive genotypes. So number of homozygous recessive upon total number of individuals present in the population, that is Rn upon N. Homozygous recessive genotypic frogs are 3, 1, 2 and 3. So 3 upon 15 equals to 20. When we add this 53 plus 27 plus 20, we will have 100%. Next one is gene or allelic frequency. So what is the meaning of gene frequency? Gene frequency means the proportion which is a particular allele occurs in the population as compared to the other alleles of that locus is referred to its allele or gene frequency. Means a particular allele, that is if we consider small allele, so what is the proportion of small a allele in the population for the given gene, it is called as gene or allelic frequency. So frequency of dominant A equals to small p. Frequency of dominant allele is represented by the small p. So it is equal to total number of capital A alleles present in the population upon total number of all the alleles of that gene. Next one is frequency of small a allele. It is indicated by Q. So number of small a alleles present in the population upon total number of alleles of that gene in the population. So we will have individuals with 2 alleles. So we will have 2 alleles with 3 genotypes. So the total number of alleles in the gene pool we have to calculate by total number of individuals present in the population multiplied by 2. Here in our population, we will have all 3 different types of genotypes, that is homozygous dominant, heterozygous, and homozygous recessive. So here total number of alleles, that is 30, because we will have 15 number of frogs. So 15 into 2 equals to 30. So frequency of capital A, that is small p allele, equals to total number of capital A alleles in the population. Obviously, we will have 8 homozygous dominant plus 4 heterozygous. So 8 into 2, 16 plus heterozygous. In each heterozygous we will have 1 capital A allele. So 16 plus 4, 20 divided by 30, we will have 67% of capital A allele in our population. Similarly, for small a allele, we will have heterozygote genotype. We have to add allele from the heterozygote genotype with a homozygous recessive genotypic individuals. So total number of small a allele are 10. 10 divided by total number of alleles present in the population, and we will have 30 alleles. So our genotypic frequency is 33%. Now we will go through one example. Coat color in pigs is determined by 2 alleles at a locus in such a way that capital YY produce yellow, heterozygote, that is capital Y small y, produce cream color, and small y homozygous recessive genotype produce white color in a sample of 1000 pigs. So in this 1000 pigs, 800 were yellow, 150 were cream, and 50 were white. So we have to calculate genotypic frequencies of the sample, gene frequencies for the yellow and white allele, and calculation of gene frequencies from the genotypic frequency. So here we will have total number of pigs in the population, that is n equals to 1000. Here we will have 3 genotypes, that is homozygous dominant indicated by D, heterozygous indicated by H, and homozygous recessive indicated by R. So D plus H plus R equals to N. We will have homozygous dominant individuals are 800, heterozygous individuals 150, and homozygous recessive 50. When we add all these 3, we will have 1000 number of individuals. To calculate the genotypic frequency, we will have formula total number of genotypes, homozygote genotypes present in our population divided by total number of individuals present in our population, that is D by N equals to, we will have 800, D equals to 800. So 800 divided by 1000 equals to 0.8. Similarly, for heterozygous H by N, we will have 150H divided by 1000 equals to 0.50. And for homozygous recessive R by N, we will have 50 for R. So 50 upon 1000, 0.05. So when we sum up these all 3 frequencies, that is 0.8 plus 0.15 plus 0.05 equals to 1. Next one is gene frequencies. So for the calculation of gene frequencies, we are very well known about that each individual contain 2 alleles. That is, for genotype YY, we will have 2 homozygous dominant allele. For heterozygous, we will have 1 dominant and 1 recessive. And for homozygous recessive genotype, we will have both the recessive allele. So number of pigs for homozygous dominant are 800. But as in our homozygous dominant genotypic individuals, there are 2 dominant alleles, that is capital Y, capital I. So 800 into 2, that is 1600. But for the capital Y in heterozygote genotype, we will have only one Y. Okay, so here we will have two alleles, but we will have only one homozygous, one dominant allele. So we will put it as it is 150. So 1600 plus 150 equals to 1750. For recessive allele, similarly from heterozygote, we will have 150 small y, and from homozygous recessive, we will have 50 individuals. So 15 into 200. So 150 plus 100, we will have 250 y alleles. Gene frequencies for the y and small y gene will be small p. For Y allele, we are using frequency indicated by small p. So small p equals to twice d plus h upon twice n. Twice d, we will have 1600 plus h 150 upon 2 into n. We will have 1000 peaks. So 2 into 1000, 2000. So we will get here 0.875. Similarly, for the Q, we will have 0.125. When we sum up P plus Q, it is 0.875 plus 0.125 equals to 1. So for the calculation of gene frequency from the genotypic frequency, our formula is D plus half H. But the frequency of capital D is 0.80, which is we have already calculated in the first table. So 0.80 plus half heterozygote genotypic frequency, that is 0.15 equals to 0.875. Similarly, for the recessive allele small a, Q equals to 0.125. So when we sum up P plus Q, it is always remain 1. So 0.875 plus 0.125, we will have 1. And likewise, for the genotypic frequency also, D plus H plus R equals to 1. We will have 0.80 dominant homozygous dominant genotype, 0.125 frequency for the heterozygous genotype, and 0.05 for the frequency of homozygous recessive genotype, and it becomes 1. So this is all about your gene and genotypic frequency. Thank you.